Welcome. In the previous module, we started covering the basics of microscopy and how it is relevant to a course or a field in field of microfabrication. We went through what are the different types of microscopes like optical electron microscopy, and then within an optical microscope what types of microscopes are available like stereo, metallurgical, inverted. I went over the details of each of these in a very brief manner. Then we went on to cover what the stereo microscope is and how it looks like, what is its utility and all. Now today we will be looking at a much more conventional microscope. This is called the metallurgical microscope. This is called the metallurgical microscope. We will look into it shortly, the actual equipment. Before we do that, when we discussed about the stereo microscope, we understood that stereo microscope works on the principle of reflection of light. But the metallurgical microscope works on the principle of transmission of light. So, we will have a sample and primarily works on the principle of transmission of light, but it can work on both reflection and transmission because of its unique construction, which we will go into. So, now let us actually look at the microscope and then understand what it is. So, we have the metallurgical microscope here. It is a very, as you can see in first instance itself, what you can make out is that this is a much more complex microscopic arrangement as compared to the stereo microscope that we had seen last time. Before I go into the details of what are the different knobs, features available on the microscope, basic thing let me introduce first. So, this microscope has both bottom illumination and top illumination. So, there is a top light source here. This black knob that is there at the back of the microscope is a light source for the top illumination. Okay? And there is an arrangement at the bottom where you have a bottom light source. Let me just switch on the bottom light source for you. See, now you can see a light glowing here. This is the bottom light source. And if I switch on the top light source, I think you might not be able to see it properly, but there is a top light source also. So, if you see, if you look at my hand, there is light coming from top also. So, these are the two illuminations that are there in the metallurgical microscope. Now, why are they important? When we saw the stereo microscope, the illumination was coming from top. So, what would happen in the stereo microscope? That illumination that is coming from the top or the light source that is coming from the top falls on the sample and it gets reflected and is picked up by the eyepiece. But in this microscope you have both options. Let us say, so same like what we saw before, we have another sample holder also here, but this sample holder is slightly more complex. It is engineered and machined for fine adjustments. So, there is a transparent platform that is kept. You can keep, if your sample is transparent, let us say if your sample is transparent like a glass slide or a petri dish or a tissue culture dish, then you can keep it on top of this and your bottom illumination can actually pass through your sample. So, that is based on transmission optics. It will work on transmission optics. So, the bottom light will pass through, after passing through certain filters and aperture adjustment equipment, it will pass through your sample and which will be picked up by your objective and your eyepiece. Now, let me get back to a few basic concepts about microscopy. The two main optics arrangements for an optical based microscope are the objective and the eyepiece. Okay, I hope you can see the objective arrangement here. So, this is a circular, this is an equipment that moves in a circular fashion. Let me switch off the light source for some time. So, this piece of part, that is part of the microscope, which we see I am rotating it now. This is called the turret. This turret can house multiple objectives. Objective is basically your core magnification part in a microscope. The objective consists of a lens of a particular magnification which is connected to your eyepiece. As we had discussed before, the eyepiece, which you can see now here. So, the eyepiece is from where you see the samples. The eyepiece provides 10x magnification. usually most of the time, and this eyepiece also provides 10x magnification. If you look at the whatever is written on the side, I think you might not be able to see, but this has 10x magnification. So, the light either transmitted or reflected light. So, the metallurgical microscope can work on both transmission mode and reflection mode. If we are using bottom illumination, it will work on transmission mode. If we are using top illumination like I showed, it will work on reflection mode, and the reflected or transmitted light from the sample is first captured by the objective. Here in this microscope or in any standard metallurgical microscope, the turret will provide for having multiple objectives connected to your eyepiece. So, eyepiece is fixed with 10x magnification. So, let us see. So, you see there is a red band here on that. This is one eyepiece, one objective, this is one lens. If you look at the lens, you can see that it is written LM plan 5x slash 0.15 and WD 10.8. WD is called working distance. See, if you can see this 5x 0.15 WD 10.8. WD is called working distance. It is like that is a measure of how close to the sample that you can get, or how far away from the sample is it useful to use that lens. It means both, and 5x tells you what is the magnification that this lens can provide. So, this is the smallest, usually this is the smallest objective lens that is provided on a microscope. It will either be 5x, 3x or 4x. So, what happens? So, this 5x lens will provide 5x magnification. Eyepiece already has a 10x magnification. So, effectively 5 into 10, you will be seeing your sample at 50x magnification. That is the overall principle. Now, suppose I adjust the turret. You can adjust the turret. As you see, the turret will move and gets locked on to the next objective that is there. Once it gets locked on, you can hear a click sound. I don't know, I am not sure whether you can hear it, but there is a click sound and the next objective comes into position. So, each objective is usually marked with a color, because as you use over time, you will just look at the color and you will know that okay, this is this magnification and this is another magnification. So, as we had seen, the 5x has red color. So, this one, the next eyepiece is 10x magnification. I think you can see that also written 10x WD 12.2. So, that 10x magnification eyepiece is yellow in color. Next one what we have is 20x magnification. You can see the next objective that is coming into position. It will keep coming into position because this is where you keep your sample. So, this is 20x magnification. So, once you put 20x, the 20x into 10x. So, it will give 200x effective magnification. Then we have two more, which is 50x here, blue one, and 100x, which is 0.8. See, if you see, the working distance for each of these lenses is coming down as you come down. This is because with a 100x magnification, you have to be very close to your sample for you to use that lens. So, that working distance will automatically come down. That is why here WD is 2.1 and the magnification provided is 100x. So, let me recap. So, we have this turret which houses all the objective lenses that are used by the microscope. You can have multiple options, and usually a turret will give for at least 5 slots to insert your different objectives. So, here in our lab we have inserted 5x which is red in color, 10x which is yellow band in color, 20x which is green in color, 50x which is blue in color, and 100x which is white in color. These colors are only for your easy use during your day to day research life. You need not go and read each lens what is the magnification. So, once we decide what is the magnification that we need, we will keep the objective lens in that magnification, insert our sample on the sample holder, and then look at it through the eyepiece. Now, the sample holder, I think you can see the sample holder. You had seen the stereo microscope before. Compared to the stereo microscope, this sample holder is a much more complex equipment. So, just below the sample holder there is a knob where my blue gloves are. I think you can see it. So, this knob is used for XY movement of your stage. This is called stage. The sample holder in a metallurgical microscope is called a stage. So, you can actually if you adjust the knobs here, you can actually move the stage. I think you will be able to see the stage moving. Now I am adjusting the x values. So the stage is moving in x direction, and I am adjusting the y value now, and it is moving in the y direction. So the illumination will be provided from bottom or top. So at that point, you cannot put your sample in the entire sample holder, because that only one particular part of the sample will come under the view of your objective or eyepiece. For that matter, it is very essential that you should be given the option to move your sample around. That is how this x y staging or movement becomes useful. Next, we have a few knobs that are below the stage. These are used for aperture adjustment. So generally in photography, or let us say microscopy also, there are few things that you look at, like photography enthusiasts among you might know there is aperture, there is exposure time, there is something called the f number, that measures the focal length at which you are capturing the images and all. Similar concepts apply to microscopy as well. So depending on how much aperture you want. So what is aperture? Aperture refers to the your hole, the diameter of the optical light collecting mechanism. So there is a knob here below with which you can adjust the aperture of your objective that collects the light. So with this you can adjust it, and you can also adjust contrast through a phase ring that is provided below. So you can adjust the contrast and work with it. So there are a lot of options provided on the microscope like this, and you need to, over time when you use it, you will get to know the exact utility of each of these options. Now let us say, let me get into the basic knobs first. So as I showed you before, this is the bottom light source. So here you see a switch here. So this is for switching on the bottom light source. So let me switch it on. It is just a rotation. I rotate it, and the light source comes into picture. I think you can see the light. See, my hands are glowing. Let me switch it off now. Switched off. Similarly, on the other side, you might not be able to see, there is another knob with which you switch on the top illumination. So I have switched on the top illumination. See my gloves, and the top illumination is on. Now, just like in the stereo microscope, we have two knobs here. These two knobs, you can see, just follow my gloves. Two knobs here. This knob is used for coarse adjustment of your focusing. So as we had seen before, the stage we can move in x and y direction, but to focus you need to move your stage in the z direction, so that your sample comes into focus with respect to the respective objective that you have used in the turret. So you can have coarse adjustment, and once you know that your sample is reasonably in focus, you need to make it sharper, right? To make it sharper, you have fine adjustment here. This is used for fine adjustment of your focusing. We will see a sample shortly and know how this is done. Then this is the eyepiece. This is the eyepiece. This eyepiece you can move around like this, so that it will suit your eye, which what is the shape of your eye, accordingly you can adjust your eyepiece. And then this eyepiece has 10x magnification, as you can see here. 10x is written here, and whatever is seen by the eyepiece is also captured by this camera. This is the camera. So this is the focusing eyepiece for the camera, and this is actually the Sony camera. This camera is connected via USB into a system. So whatever we are seeing, we can actually capture it through a software that comes along with the microscope. So if you see the screen, you have a software provided. If you see the screen, you have a software provided to capture the images. It is a very basic software. You just see the sample under your view. There is a capture option here. You just click it, capture to a file, and then just capture it, and you can save it as an image like this in your desktop. or in your drive, and you have options to do white balancing for your image, and you can change your red, green, blue saturation levels in the image, and you can adjust the resolution at which you want to capture your images, and like auto exposure you can do, so that it adjusts for that also. So many things are there. Let me take it back to the default values. Okay. So I have done white balancing. So this is the software which you use to capture the images. Now let's get back to the microscope. So as you had seen, this is the light source for the bottom top illumination. This black color square that is seen at the back side, that is a light source for the top illumination. So there are few knobs here. See, so there is this is again, there is a piece here, I hope you can see. So this is for adjusting your aperture again. How much of your source light actually falls on your sample. So if you want to adjust that, you can actually, so that is not basically why this is used for. That is also a good utility, but more importantly this can be used for putting filters, like color based filters, like red filter, green filter, blue filter, etc., which you can put some filters are there, some filters will be there which you can insert here. Then you have one more similar arrangement for multiple filtering here. See, it is a similar one, but just that it is a smaller radius. Here if you can see, we have put a filter here. I think you can see a small color difference. So that is one filter that is put. So these filters you have to insert. There are places where you can insert these filters into the microscope and they will come in picture. So suppose you want to do fluorescence imaging. So what is fluorescence? Let us say you are studying some sample. So I will show you a few more filters which we are having. I thought, because anyway we went to the filters, we thought we will show you. So let us see, this is a yellow color filter. I don't know, through the camera it might come as a different color, but this is a yellow filter. Likewise we have a blue filter, green filter, blue filter. So this is blue filter, this is green filter. If you can see, it is written IFB less than or equal to 480 nanometer. This is the wavelength of the light. So this is like it is telling you that it will pass anything below 480 nanometer, and that is blue light filter. So as you know, if you are aware of optical microscopy and energy related relations, so basically blue light has more energy than yellow light. Yellow light goes closer to the infrared spectrum. Blue light goes close to the ultraviolet spectrum, and blue light has lower wavelength. Yellow light has higher wavelength. If you know Planck's law, the wavelength energy relation is h nu, where nu is the frequency, or h nu is also equal to h c by lambda. So as lambda goes up, your energy comes down. Similarly, as lambda goes down, your energy goes up. Blue light has low lambda, 480 nanometer, less than 480 nanometer. So blue light has higher energy. Yellow light has higher wavelength, so it has lower energy. Remember, h c by lambda. H is Planck's constant, c is speed of light, lambda is the wavelength of the light. So here if you see, this is green filter. Again, so green falls in between blue and yellow. So as from your school days you might be studying about VIBGYOR, violet, indigo, blue, green, yellow, orange, red, VIBGYOR. So this is green, 520 to 570 nanometer. You can see, right? Similarly we have blue, less than 480 nanometer. Likewise you can use these filters, insert it into the corresponding slots in your microscope and use it. So when we tell this, we need to discuss about fluorescence microscopy. There is a subclass, fluorescence microscopy. So there is a subclass of microscopy. What happens in that? Let us say we have a sample and we add a particular chemical to the sample. What that chemical does is, it goes and interacts with some particular feature of that sample in such a way that after interacting, that particular sub feature of the sample emits a particular light. So, it starts emitting light. What is fluorescence? Fluorescence is when a substance starts emitting light. So, when it emits light, we can capture that. So, let us say there is something called nuclear staining of cells. So, let us say we have a petri dish containing lot of cells, and then we want to study how the cells nucleus, you all know nucleus is the core part of a cell. We want to know how the nucleus is behaving. What we do is there is a chemical or a dye, it is called a DAPI. We add that chemical to the cells. What this DAPI does? DAPI goes into the cell, goes into the nucleus, binds with the DNA that is there, and once that binding event happens, it starts emitting blue light. So, then if you put this filter for a blue light, let us say fluorescence microscope and a filter if you use, without using any illumination, after adding the chemical, because this sample itself will act as a light source. Why? Because this is fluorescence. So, you add a chemical to the sample reacts with the chemical in a specific manner, and it starts emitting light from specific regions within the sample. Then what happens? It emits light, that light is captured. When you look at the image, you will see light coming out from only where the nucleus is present, because that chemical DAPI went and interacted only with the nucleus of the cells. So, in the image you will see blue color dots throughout your field of view, and those blue color dots will correspond to the nucleus of your cells. This is the basic method in which fluorescence microscopy works. Fluorescence microscopy can also be done with this microscope. We will not be getting into that much detail because that is a more biology oriented thing, and this is a microfabrication course, but we thought that you should know about it. So, this microscope, but right now also it is capable of carrying out fluorescence microscopy also. So, as I told, there are filters and there are aperture adjustment equipment like this. We can adjust the aperture like this and then conduct the microscopy. Now, we will look at a few samples, 1 or 2 samples, and see how bottom illumination captures images, how top illumination captures images, etc. We will see a particular sample and how we are seeing the image through the microscope. We have there is something called samples basically. So, these are this is a box that comes, this is readily available that we can purchase, and this will have representative samples of different cell types. So, these are slides that are all are inactivated, nothing to worry about. These are used for us to study. This is purely for academic purposes. So, let us say this is histology of mammalian cells, and this is cardiac muscle histology. Can you see this? Cardiac muscle histology. Let's see one more. Just I will randomly select. We have Entamoeba histolytica, which is a protozoa. Okay. Today let us see, I have taken one sample from here, and that is inactivated malarial parasite, because all of you are aware of malaria disease. So, I thought we will show you malarial parasite. This is basically inactivated, nothing to worry about, you will not get any disease with this, but you have to always take precautions because you are handling biology related topics. Okay. So, now once we put the sample, we have to first see it through the eyepiece. Because we have a camera connected to the sample, you can actually see what we are seeing on the slide in the screen. If you look at the screen, you can see. Now, you can actually look at only the screen as I change the different knobs. So, let us say now you can see, right, there are a lot of dots and other bigger pieces. So, this is the malarial parasite slide which we are seeing. Now, let us say I am moving it in the x direction. So, as I move it in the x direction, the slide will also move, and you can actually see how it is changing, how the view is changing and all you can see. Now, let us say I move it in the y direction, what will happen? See, it is going down. So, you can see the different parts of the cells of the parasite, what are there. You can see so many things. Okay. Now, let me adjust the focusing. So, if I adjust the focusing, right, this will go out of focus. Now, you will not be able to see much. See. Now, you have to adjust the focusing so that you are able to see what you are intending to see. Yes. Now, I have used 20x magnification, like that you can use the 10x one, so that you will get a much more macroscopic view of things. You can see the things changing. This is a microscopic view. Now, suppose I go to 50x, it will be a totally different view. So, this is a 50x image of the same. Oh, now let me take it into focus. Okay, I have kept it into focus now. Now it is in focus. Like this, you can go in and try this. Now I have used bottom illumination. See, the light at the bottom is blinking and top illumination is not used. Suppose I switch on top illumination, I can even have both illuminations at the same time. See, the image is getting brighter. I have used top illumination. So, and you can even have only top illumination. Let us say I switch off the bottom illumination and you work with only the top illumination. So, that time, see, the image seen is slightly different, because we have used a blue filter. Let us say I remove the blue filter, you will get white color. So, I have removed, we had this blue filter kept. So, let me put it back, then you will see again that image has become blue. And that too, how, see, you are seeing a circular image, right? That is because you are put a filter like this, circular filter. Then now let us see one more thing. So, now you have understood how top illumination works, bottom illumination works, how we have to focus it, how images will be there, how image will be captured, as I have shown. Now, two more things we let's introduce. Just like the filter for the top illumination, we have filters for the bottom illumination. These are the filters for the bottom illumination. Let us look at the names of it. So, let us say we have IF 550. So, intermediate frequency 550. So, it is like it represents the wavelength at which this light will be filtering, like what light effectively will be going to your sample. Like that, this also has a name called LBD. So, let us put LBD in this, then see what happens. And then we have, once we are putting it in the bottom illumination, we have to switch on bottom illumination, right. So, we have switched on bottom illumination. This is another light source, another method by which we can do fluorescence microscopy, which I discussed before. What happens in this will be, you excite your sample with a particular light. So, your sample will respond to only a particular wavelength. Let us say you excite your sample with blue light only, then parts of the sample which will respond to that blue light will start emitting another light back. So, that way you can measure fluorescence microscopy. So, now I have put the blue light filter. Now let me remove the filter, you will see the difference in the image. So, it has become it is white now, like bright field image. Then let us put the blue light filter, see, it has become blue. Now let me put the next filter, let us see what happens. This is green light, see, it has become green. So, this way you can do lot of things with microscopy. Microscopy in itself is a very broad field, we are just giving you a glimpse of it, and which is only required for you to characterize your sensors and your devices, like MEMS devices. So, this is basics about metallurgical microscope. Now, we will be covering the inverter microscope also. So, you get a wholesome understanding about the different types of microscopes that are available. Thank you.